Good day, everyone, and welcome to another episode of our Clinical Trials Unboxed for November 2024. My name is David Taylor. I'm the VP of Research and Strategic Partnerships at ALS Canada. We have a very special guest with us today who is here from Ionis Pharmaceuticals, Dr. Stephan Miller, the Director of Clinical Development, and who has worked in industry for over 18 years, is here to tell us about the FUSION clinical trial. We're very excited about to learn more about this, to be able to tell you about it, and then to see how we might be able to support getting this trial to the point of results. But from all of that, I'm going to pass it over to Dr. Miller. The floor is yours, and please do enter any questions into the chat or into the Q&A if you have them, and we'll be happy to have a little discussion afterwards. So the floor is yours.
Thank you, David. Yeah, thank you very much for the opportunity to talk today about FUS-ALS and our FUSION study. So FUSION is a randomized, double-blind, placebo-controlled trial to evaluate a drug called Ulefnersen as a potential therapy for FUS-ALS. Here's my disclosure. I'm an employee and shareholder of Ionis Pharmaceuticals. So FUS-ALS is ALS caused by mutations in the fused in sarcoma gene. It's very rare. It's about 1% of all ALS, and it can occur both in familial and sporadic ALS. So this figure here shows with the calendar year going up vertically, and then across horizontally, it shows the different genes that have been identified to be involved in ALS. So SOD1 was one of the first ones. FUS-ALS is here in the middle, and the size of each circle corresponds to how common ALS caused by that by mutation in that gene is. So FUS, the little purple circle in that red rectangle there in the middle, much less common than SOD1 ALS, maybe 1/4 or 1/5 as common as SOD1 ALS, and much less common than ALS associated with changes in C9orf. The frequency varies across populations, and in most Asian countries, it's 2 or 3 times more common in Asia than it is in the US or in Europe. So an interesting thing about FUS-ALS is the age of onset is highly variable. I'd say FUS is probably most well known for affecting younger patients. The average age of onset is late 30s or early 40s, so generally earlier than the overall ALS population. But the range of onset varies incredibly from individuals as young as 11 years old to individuals as old as 80 years old. So FUS-ALS has, I would say it's as heterogeneous as ALS in general in terms of age of onset. Mutations in FUS are the most common known ALS causing mutations in early onset ALS. So over 40% of juvenile ALS, over 50% of pediatric ALS. If the mutation is known, it's most likely to be a FUS mutation. The rate of progression of FUS-ALS is highly correlated with the age of onset and the particular mutation that the individual has. So early onset cases tend to have rapid progression and often short survival of 12 to 18 months, and patients with mid to late age of onset have a progression rate and survival more similar to the overall ALS population. The types of symptoms initially are also very heterogeneous. They may be bulbar, so problems with speech or swallowing. They may be axial trunk or neck weakness, or may affect upper limbs, lower limbs, or there may be respiratory symptoms. So what is FUS? FUS is a DNA and RNA binding protein that regulates steps in gene expression, so turning the information from the gene into a protein. So FUS is involved in transcription, splicing of messenger RNA, a messenger RNA transport. So this little picture is a schematic of the FUS protein, and it shows all of the different, well, it shows many different places in the FUS protein where mutations that cause ALS can occur. Many of them are in this part on the right where it says NLS. This is the nuclear localization signal. So in order for FUS to do its jobs, regulating, turning genes into RNA and turning RNA into proteins, it has to be able to get in and out of the nucleus. So from the cell body into the nucleus, and if there's a mutation affecting this nuclear localization, that localization is disrupted and it causes FUS, the protein, to aggregate or clump in the cell body, in the cytoplasm. And that somehow leads to motor neuron degeneration, and it's known as a toxic gain of function. Those little clumps of the FUS protein are somehow toxic to neurons. There are many different mutations in the FUS gene, over 200 variants, and more than 40 of them are known to cause ALS, mostly by affecting this nuclear localization process. So here's the drug that we're testing, Ulefnersen. Here's the therapeutic rationale and how we think it works. So Ulefnersen is a type of drug called an antisense oligonucleotide. It binds to FUS messenger RNA and causes it to be broken down by an enzyme called RNase H1. So in the picture here, FUS pre-messenger RNA is orange. Our drug is in the purple. So the Ulefnersen ASO binds, and that duplex together is broken down by an enzyme that's normally in cells called RNase H1. So when the RNA is broken down, no protein can be made, and so none of that mutant protein can be made, so it shouldn't clump in the cytoplasm. The chemistry of this drug, Ulefnersen, and the way it works are very similar to the drug tofersen or Qalsody, the drug that was approved last year for SOD1 ALS. So our therapeutic hypothesis then is that degrading that mutant FUS RNA will reduce the amount of protein that can aggregate and will reduce that toxic gain of function so that motor neuron degeneration will be reduced, and that will slow or stop progression of FUS-ALS. So this slide is the story of the first person who was treated with Ulefnersen. It's the story of identical twins, Alex and JC Hermstead. They both had the same P525L mutation. This is one of the mutations I mentioned characterized by early onset of ALS and fast progression. One sister, Alex, had symptom onset at age 11, one of the youngest reported cases. She was diagnosed with ALS at age 16, and she died about a year later. I mentioned how heterogeneous this disease is. Even among these identical twins, her sister JC did not have onset and was not diagnosed until age 25, 14 years later. She also had rapid progression, and she was the first person treated with Ulefnersen by Dr. Neil Schneider at Columbia University. So her family was aware of FUS-ALS. Dr. Schneider was following JC pre-symptomatically, and he worked to accelerate the entry of Ulefnersen into the clinic. So JC's treatment began about 6 months after symptom onset, but unfortunately, after she had already experienced significant disease progression. So in ALS, as you likely know, there's a rating scale, ALS functional rating scale. and it's shown in this figure here. So when she started Ulefnersen treatment, she was already pretty significantly progressed. And Ulefnersen was associated with an apparent slowing of her decline, but she did die about 18 months after disease onset and about a year after her initial treatment with Ulefnersen. So it's not possible in the context of this open label single patient treatment to know what would have happened, if she had been on a placebo, for example, how rapidly would she have deteriorated. So the really great contribution of JC and her participation in this program is that she and her family donated her tissue to science, and examination of the autopsy tissue of her nervous system showed that mechanistically the drug worked as expected. So the drug was broadly distributed through the CNS and caused a very large reduction in that mutant FUS protein. And also the way the cells and the tissue looked under a microscope, the histopathology was greatly reduced after Ulefnersen treatment. So this provided very important information for starting the fusion study. And the objective with the fusion study was, rather than to have Dr. Schneider treat a few patients like this with open label, to really fully evaluate Ulefnersen in a placebo-controlled way. And if it is found to be effective, then to be able to make it available to all patients with FUS-ALS. So here's a slide on the fusion study design. Fusion is a phase 3 study. It has a 72-week placebo-controlled double-blind period. And at the beginning of that period, participants are randomized 2-to-1 to Ulefnersen or placebo. So 2/3 chance of getting the active drug during that controlled period, and 1/3 chance of being on placebo. After completion of that randomized period, patients who wish to continue will all get open label treatment, and Ulefnersen is available to those patients for up to 5 years until hopefully we are able to be successful, and if we find that the drug is effective, be able to get it approved by FDA and Health Canada, for example. So the way this works is that the study drug, Ulefnersen or placebo, is administered by a lumbar puncture or spinal tap every 12 weeks with an additional loading dose after the first 4 weeks. So to participate in this study, participants need to come to the clinic for each of those drug administrations, and then there are additional remote contact visits between the clinic visits, so either by phone or by video. Many assessments are involved in this study. The functional rating scale, of course, ALS, FRS, measurements of breathing capacity, the slow vital capacity, measurements of muscle strength with handheld dynamometry, and a number of different questionnaires that measure different things about physical function and quality of life. This is the first controlled study of Ulefnersen, so establishing safety will be a very, very important part of this. And there are many safety assessments in this trial. Safety lab samples are taken of blood, urine, cerebrospinal fluid for safety labs. All adverse events are followed, and there are also MRIs for imaging and ECGs. And the size of this trial is small compared to most ALS trials. Planned randomized population is up to 85 patients. Here are some of the key eligibility criteria. The eligibility for this study, very, very broad. It's very rare. We want to test the drug in as wide of a range of FUS population as we can. Of course, there has to be an approved mutation in the FUS gene, one of those pathogenic or likely pathogenic mutations. Very broad age. Patients as young as 10 years of age can participate with no maximum age. A participant, of course, would have to have signs or symptoms of ALS, and they cannot already have a tracheostomy in place or already require ventilation more than 22 hours a day. Stable background therapies are allowed. For example, riluzole is allowed. Tofersen, that drug for SOD1 ALS, is not allowed because uh, it's not anticipated that ulefnersen would be helpful if a participant's ALS is actually caused by a SOD1 mutation. And then here's a link to the record for the trial on clinicaltrials.gov that gives a lot more information, including a complete list of all the eligibility criteria. The study's active now. There are 24 sites in 16 countries. So Canada, US, Europe, three countries in Asia, and also one site in Brazil. And enrollment began mid-2021 and is expected to complete in early 2025. The site in Canada is Dr. Genge's site at the Montreal Neurological Institute, and they are enrolling now. We have a lot of resources to offer. The trial supports patient travel. So if a participant doesn't live near a site, expenses related to the participant's travel to a site for them to travel and a caregiver to travel can be supported by the study. So flights if needed, hotel stays, meals, things like that within reason. And interpreters are also available during clinic visits if needed. And then the final part of this, I'd like to point out that outside of this trial, Ionis sponsors an ALS genetic testing and counseling program. This is a free program, so particularly helpful if a testing like this is not covered by your insurance. It's not just for FUS. It's a 33 gene panel done through Prevention Genetics. The eligibility are shown here. Suspected or confirmed ALS with or without family history, person of any age, or if a person has a family history of ALS, but they don't have symptoms yet, they are also eligible if they are adults. And along with this testing program, we also offer genetic counseling, so both pre-test and post-test genetic counseling, both for patients and their families. So I think that does it for me. Some links here for more information, the clinicaltrial.gov registry, an email and a phone number for patients to contact who are interested in this study, and then a link for the Ionis sponsored genetic testing program. So thank you, David and ALS Canada, for the opportunity to present this study. Many thanks to many other people, primarily J.C. Hermstad and her family, Dr. Schneider at Columbia, and of course, all of the participants so far in our trial and all of our investigators and staff. Thank you.
Thank you very much, Dr. Miller. That was wonderful. And this is exactly why we wanted this to be here, because again, we're hopeful that the word getting out can continue to make sure that everyone has an opportunity for genetic testing. This is offered obviously through the panel, and then other ways that people can get it through their clinics to make sure that if anyone does have a FUS variant, that they will get identified and potentially have an opportunity to be involved in the trial. I did have a couple questions. One of the questions is around the different types of FUS. So often clinicians are, and I know you alluded to this in your presentation, but often people talk about FUS for the really aggressive younger individuals to look out for that specifically. But there are other things that you kind of highlighted there that are sort of more indicative of individuals who may have a FUS variant that could be, someone that you would want to expedite for or look into genetic testing. Would you be able to sort of maybe mention a little bit more about some of those heterogeneous things that present in people with FUS variants?
Sure. Yeah. So I think what you're getting at is the most remarkable thing, the thing that FUS-ALS is famous for, is that it affects very, very young patients, patients as young as 11, which is so extremely rare for ALS. But that's so rare. That doesn't mean most patients are young. Most patients are uh, older, patients in their 40s, 50s, 60s, or even 70s. And unfortunately, the presentation is also very, very heterogeneous. Um, Things that are common in FUS ALS are uh, um, axial presentation, neck weakness, bulbar symptoms, problems with swallowing, but any type of symptom can be the initial presenting symptom. So just another reason why it's so valuable to have people get genetic testing if they want to.
For sure. And so one of the interesting things about this trial too is that it was mentioned that it's very broad. So someone who is in their teens who may have a rapid progressing form of FUS would be eligible for the trial, but someone else who might be older and maybe has a less aggressive form would also be eligible. When it comes to sort of getting the results, is that sort of indicative of a hope that this would be a more robust effect that you'd be able to see even with that level of heterogeneity, a level of potential efficacy? I know you alluded to the fact this is the first time it's been going widely in people, so obviously safety is a big piece of this, but the hope that there could also be an efficacy signal would be, one would think, quite difficult with such a heterogeneous population within the trial. So would you be able to speak a little bit about that?
Sure, sure. Yeah, it's a difficult task to have a very broad inclusion and a small study. Um, so what we hope is that the drug will work well for everyone, and that won't be a problem. But in our statistical analysis, we're doing some things at the beginning and some things at the end to help us with that. So one thing at the beginning is people are randomized 2-to-1, but within certain characteristics. So fast progressors and young patients are randomized 2-to-1, and older
See.
patients and slow progressors are also randomized 2-to-1 to help us prevent having an accidental imbalance between, maybe all the older participants were on placebo, and then, a disproportionate amount of the younger ones were on Ulefnersen, that type of thing. So there are things in the way the trial is set up to help with that. And then we have to pre-specify our statistical analysis plan. So when we look at the results at the end, we will also be able to look at subgroups. So patients that are young, patients that are older, patients that have a particular mutation. So we hope that efficacy is great for every subgroup, but we'll be able to
Sure.
look at that more carefully at the end.
That's great. Thank you for clarifying that. For sure, that I think is fantastic. And I think, one of the things that would be great for, sort of a lay audience out there as well, is the reason why we would talk about that more robust effect from a scientific standpoint is, unlike a lot of other therapies that are out there, this one is probably most akin to tofersen, as you alluded to, which
Mhm.
we have seen through years now of experience in at least largely trials and open label extensions, but now some real world sort of anecdotes that this is, when you know what the target is and you know that there's a gain of sort of toxic function, you can reduce this. It seems to have an effect in SOD1 ALS, and because in FUS ALS there's that same sort of scientific backing, the hope is for looking for something a little more robust. So I mean, I think it's very exciting, and we certainly want to see that get enrolled. So, can I ask you another quick question? And please, if anyone has any questions, feel free to add them into the Q&A. I'm happy to answer or ask those. Um, so with tofersen, for example, when it did get to the point of being able to demonstrate efficacy through the trial with the open label extension and further trial studies after the extension of Valor, they ended up then putting in something like Atlas, which was really the early testing of an individual's based on the biomarker elevation of neurofilament light. Um, if you were to see something quite robust in FUS, do you expect that there would need to be a similar sort of trial? Do you know if there would be at least a drive to start to look at avenues to look at earlier treatment with this if there was efficacy that could be proven post-diagnosis?
Yeah, it's something that we would consider, but keep in mind, the Atlas trial is a very difficult trial to enroll and conduct. And now here we're talking about FUS 1/4 or 1/5 as common as SOD1 ALS. So it would be challenging, definitely,
Yep,
to do that. Um, in this trial, we allow for enrollment signs or symptoms of ALS. So a patient, a potential participant does not already have to have a definitive ALS diagnosis as long as they have a pathogenic mutation. So we will have a subgroup in this trial who start out with very, very, very good functioning, and that will uh, hopefully provide some evidence as to whether it will help early on, even pre-symptomatically.
that's incredible. Actually, I mean, I don't think there's a trial out there that's doing as much with 80 plus participate 80 to 90
Yeah,
participants as this one in terms of different levels. But I mean, it's a great testament to what has to be done if you want to treat rare groups. And I mean, I think it's also great that you highlighted the Hermstads and I'm not sure if you're able to talk about, but I think that that also was sort of stimulatory for many more studies that will be like that, where an individual can be tested at a sort of an N of one level, an individual level, and that can be able to help people where there may be very rare variants in not just ALS but other diseases.
yeah. Yeah, there's an organization called NLOREM, N-LOREM, that was started by the former CEO of Ionis, Stan Crook, and it does exactly what you just suggested, and not just in ALS, but in many other indications, and trying to find N of one treatments for conditions that are much, much more rare than FUS-ALS, things that would not really make sense for a pharmaceutical company to go after because it would be hard to, get it
Of course.
any kind of return on the investment in research.
Yeah. That's such an incredible story from beginning to now for Ulefnersen, and I know you're the project team leader for Ulefnersen, so I'm not sure if you can sort of riff on anything else going on at Ionis, but I know obviously, and many people know that Ionis has had a good long-standing partnership with Biogen in advancing ASOs for ALS. It is the ALS gurus for basically everywhere. And I know there's a lot of preclinical work that's being done
Sure.
in ALS in partnership with Ionis. I was wondering if maybe you could riff on, sort of whether there are other things you have that you're thinking about in the pipeline to continue working on the disease in different ways.
Sure. Yeah, ALS is an active area of research at Ionis, and there are a few things that are in very, very early stages that haven't been talked about publicly yet, but they might be at an early stage where it's like, this looks like a good target. This looks like a gene that's involved in ALS, and we, have certain criteria for like, are we able to make a drug that could address that mutation? And do we have a good way of testing whether that would be effective, like in cell culture or an animal model in a rodent, that kind of thing. So we have some very, very early stage things like that, or where the target has already been identified and agreed upon, and now it's just a matter of working on many, many different iterations of that ASO to find the one that works uh, best on that mutation, and then consider whether that could move ahead into animal testing or into a phase 1 study. So yes, still an active program here at Ionis, but no other study that's about to enter the clinic that we can talk about.
Well, this is certainly a big one anyway to have going right now. And certainly, if there are scientists out there, young scientists that have ideas or great targets, I know that reaching out and seeing if there's any avenues for collaboration is always something that's an option. And I want to say thank you again. I will tell everyone out there that if you want to learn more about Ionis Pharmaceuticals, there's a great podcast called Hope Lies in Dreams. It really talks about how antisense oligonucleotides almost didn't make it to where we can have trials like the Fusion trial and things like tofersen and amazing therapies for many other diseases like SMA. Check that out. It's an incredible story and company behind it and a big commitment to ALS. So thank you very much for being here, Dr. Miller. And thank you everyone for being at this episode of ALS Clinical Trials Unboxed. And we'll see you again for the next one. Take care, everyone.
Thanks for the opportunity.
Thank you.
Take care.
Bye.
Bye.